And so I'll introduce Adrianne, who is now a lecturer at the Queensland University of Technology. So as she mentioned, it's 11:30 and we're really happy that she's staying up all night. It's a real dedication. And she's going to talk about using data to inform decision-making in cancer therapies. And we're excited to hear. So thanks for having me.
Yeah, so thanks everyone for joining us in whatever country you're in and whatever time zone. So I'm going to give a bit more of an introduction talk as well that's talking about data fitting and just data in general and how important that is to understanding cancer therapies and helping improve our treatment of cancer. So to start off, I thought it'd be good just to talk about what I find interesting and challenging about modeling cancer, which is the fact it occurs across so many different scales. So when we consider a cancer or a tumor in the body, we might consider it to be in, say, one organ of the body, or it might have metastasized to multiple organs. But that means that you can model it in many different ways. For example, some people might be interested in modeling how drug that might be injected into the veins distributes throughout the body and how much of it arrives at the tumor. And so in that case, they might consider a full body model or a PK-PD model or a pharmacokinetic pharmacodynamic model, or you might be more interested in what's going on really locally within the tumor once you apply a treatment. Once you actually get to the tumor site, what's going to happen to these cells? And like Morgan just spoke about before, if you do have a subpopulation of cells that are resistant, how is that going to impact the outcome of treatments? On top of that, a lot of the talks over the next 3 days are going to mention how involved the immune system is in response to cancer and in response to a treatment. And so in that case, sometimes you're going to want to model right at the tumor site what's going on with immune cells that might either be helping promote the growth of the tumor or actually doing the killing of the cancer cells. And lastly, a lot of people actually are now turning to look at modeling what's happening within a cancer cell. That has a lot to do with treatments that are now being engineered to target the DNA of a cancer cell. And so to be able to optimize these treatments, we might want to actually consider what's happening directly within a cell and how these treatments might be able to induce cell death. And so really modeling cancer is a big challenge and it occurs across many different scales. And there's a lot of really great modeling works out there. And I'm going to try and talk about two kind of simple works that we did to try and tackle the problem of understanding and helping improve cancer treatments. Because this talk is focused a lot on data, I thought just to do a general intro, here's some of the data that you might encounter as you're modeling cancer or might have already encountered. So one of the first places that experimentalists start when they're investigating a treatment is at the in vitro stage. So when we say in vitro, it means outside the animal in the lab, usually in a petri dish or in some other form. And so a lot of these in vitro experiments you might think are not necessarily useful, but in fact, they can be really useful to a mathematician. So this is an example of an in vitro experiment here, where what our collaborators did was they measured the cell viability across many different doses of a drug called PAC1. So PAC1 is a type of cancer drug. And in this case, they were injecting the drug into a cell dish with ovarian cancer cells. And so what they're measuring here is as you increase the dosage, how many cells or what proportion of these cancer cells are left remaining. So unsurprisingly, as you can see, as you increase the dosage size, the number of cells or the cell viability decreases quite significantly. So the drug is definitely having an effect on the cells. However, there are low doses where the drug doesn't really seem to impact the cells. So this kind of allows us to fit kinetically what's going on when the drug is actually inducing death into these cancer cells. And so from a data set like this, we can obtain a mathematical model for how the drug, PAC1 in this case, is inducing death in ovarian cells. And so you can see here, our fit is in gray to the blue data, which is the mean and standard deviation. So in vitro experiments are really useful experiments for when you're trying to fit subparts of your model, or at least calibrate aspects of it to data. Another large part of data that you'll come across is in vivo measurements or sera measurements. Sera meaning in the bloodstream or in the plasma or in the lymphatic system. So a lot of the time when you look at dealing with in vivo data, you're probably going to be looking at mouse-based data. And so this is a really good example of an in vivo data set here where I believe there are 6 mice here. They've measured the tumor volume, which I've converted to number of tumor cells over time. And I believe this isn't under treatments. This is just a tumor volume growing over time, over 55 days. And so these types of data sets are really useful because they allow us to calibrate, for example, tumor growth, which is what Morgan spoke about and introduced in her talk earlier. And so in vivo data, just like in vitro data, can be really fundamental to a mathematical modeler and really useful to calibrating parameters. And on top of that, sero measurements as well give us a picture of what's going on at that full body level. Even though we're considering a mouse, it still allows us to kind of extract what might be happening if we considered a human at the full human level. And this is measuring the, it's a SEEP, which is a kind of a marker for cancer. This is measuring that marker in the bloodstream, or the plasma, sorry, over 14 days after the first plantation of the tumor. And then the last type of data that you might encounter is actual clinical trial data. So this is an example of a clinical trial for the only oncolytic virus, which I'll talk about in a little bit. The only oncolytic virus that's been accepted as, or been approved, sorry, as a treatment for cancer. And so this is measuring the overall survival of a clinical cohort, so actual human patients, over a certain number of times, so 55 months in this case, under these two different types of treatment, so TVEC and GM-CSF. And so what you can kind of read out of this is that patients on the TVEC treatment, so the blue arm, had a higher overall survival rate compared to just the GM-CSF treatment alone. Now, this data might not seem as useful as the others in terms of parameterizing a model, but it can be really useful for, I guess, checking your model and making sure that it actually conforms to our understanding of how effective a treatment is. Now, these are just some very basic examples from these different areas, but I thought it might be useful to see these if you're kind of just getting started in cancer modeling and you're interested in the different types of data that you might encounter. And so I also want to talk very briefly as well about some very simple data fitting methods. And I'm going to talk about my favorite, which is a simple least squares fit. And so the way data fitting works usually is that you're going to consider you've got a model, which I've denoted here as y of tp. So our model is defined for some time t and for parameters p. And now the goal usually when you have a model is to obtain an estimate for these parameters, or this vector of parameters that I've called p here. And in saying that you're wanting to obtain an estimate, the goal is really to minimize the residual or the difference between the data, so some set of data that you've got, like the ones I showed on the previous slide, and the model at the specific data time points. And so this can be just written simply as the distance between these two, or you might actually want to look at minimizing the L2 norm, which is what least squares is all about. So to kind of show what this all means pictorially, if you consider you've got this data here, which is these stars, and I've kind of labeled this is data point from T1, so time 1, and this is data point from time 4. And then I kind of plot my corresponding model output, which is a continuous line because the model I'm using in this case is continuous. So you can see in some places it's able to approximate the data pretty well, but in some places it seems to be missing. So when we're trying to do data fitting, what we're trying to do is take what our model's value is at the equivalent data time points and try and minimize this distance here, or this difference here between the data and our model approximation. And so one thing to point out is that data fitting is really hard. I think that we all start data fitting and think it's going to be really easy, and then it actually turns out to be quite challenging. So don't ever feel frustrated or disheartened if you start trying to fit a model and it just doesn't work. There are lots of different methods to kind of try. And also it's not bad to go back to the drawing board and ask whether the model actually matches what's going on biologically, because sometimes that might be why the model's not fitting. So I'm going to start off by talking about an example of where we've applied data fitting to a cancer treatment. And that cancer treatment is known as an oncolytic virus. So to understand an oncolytic virus, you kind of need to understand the virus life cycle. And as we all know, now we've lived with a pandemic or been in a pandemic for so long, we're all quite interested in what's going on in a virus life cycle. So if you consider you've got a population of uninfected cells that are proliferating, when a virus comes along, it will infect these uninfected cells and it will hijack the machinery within the infected cells and start to replicate. After a while, the infected cell will then burst, releasing all the new virions or viruses that were created, and these viruses then go on to infect other uninfected cells. And this process just goes on and on and on and on. until your immune system comes in. So our immune system is a fantastic, wonderful thing because it's able to very effectively, normally, come in and clear off a virus. COVID obviously is posing a bit of a challenge for a lot of people, but predominantly when we get a viral infection, the immune system is able to come in and it kills off all the free virus particles as well as any virus infected cells. What's exciting about viruses, or why we care about them in the cancer context, is because we can genetically engineer these viruses so that they are not able to infect normal uninfected cells, but preferentially infect tumor cells. So by doing that, what we end up having is that the virus infects tumor cells, it replicates within them, and then actually ends up killing them. And then at the tumor site, you've got these infected cells that create new virus that then goes on to infect neighboring uninfected tumor cells, and the cycle keeps going. The challenge, however, is that the immune system is programmed to remove a virus. So the immune system will come in and try and clear off any free virus particles or infected cells. However, not too recently, but more recently, the idea has become that we can actually harness this immune response that's programmed to kill off the virus to then target any tumor cell it encounters. And so this is kind of known as viroimmunotherapy or oncolytic virotherapy and immunotherapy. And so I'm going to talk about one type of treatment that's kind of an example of this that we've been working on and trying to help optimize for patients. And so that's known as dual oncolytic virotherapy. So this work was done with a few different collaborators, Mary Claude, Tyler, and Morgan, of course. And the idea here was to take these two viruses and consider them as oncolytic viruses, so programmed tumor killing viruses, and then to determine whether we can figure out what the optimal treatment strategy would be if we were to actually treat a human cohort. So we had two main questions that we were focusing on. We've got these two viruses, one's called vaccinia and one's called vesicular stomatitis virus, or VSV. And we wanted to know how many injections of vaccinia or VV should you be giving a patient? And then how many days do you need to wait before you give the VSV injection? So these might seem somewhat simple, these questions, and they are to a mathematician. That's the beauty of mathematics, is that we're able to formulate a model that can actually attempt to answer these questions. But experimentally, it might be quite hard to determine what the answer is because you might have to test multiple different permutations of these two viruses. And so that was our goal. So we started off by building a mathematical model for these two viruses in a tumor setting. And so we use this system of distributed delay differential equations here on the right. I'm not going to go through the equations in too much detail, but I will just briefly talk you through the model. So what we started off with was considering we've got a tumor population that's growing. And we called these cells susceptible because we imagine that they're susceptible to the virus infection and they're susceptible to immune cell killing. And so these susceptible cells are proliferating and they're being modeled as going through the cell cycle. So you've got cells that enter quiescent stage, they enter GAP1, which means they're preparing to undergo division. They actually then enter mitosis, so they actually start to divide and then go back into quiescent mode. So then they've already divided. And now, just like Morgan spoke about before, we also considered that a subpart of the tumor might actually be resistant, so might be resistant to treatment. In this sense, we considered it to be resistant to immune cell involvement because we know that's a major issue in cancer treatments, that the immune system just doesn't seem to recognize that there's a tumor inside the humans. And so then what we wanted to do was build on top of that the two oncolytic viruses we considered, the VSV and the VV. And so we modeled a population of tumor cells that were infected with either virus, and then also those two viruses themselves. And so we had that the virus was able to infect tumor cells that were proliferating, create these infected cell pools that then died and created new virus that then went on to continue the process. And so like I just said, we had two questions. We wanted to know how many VV injections do you give and how many days do you wait between the VV and the VSV injection? So to do that, we decided to take a virtual clinical trial approach. So what that means is we wanted to create a cohort of virtual humans that we could then simulate all the different permutations of these treatments on. And so to do that, what you usually do is follow this kind of process here, where you take a disease that you might be interested in and you create a model for it, whatever model you might be interested in or might like using. And then from that, you extrapolate out a population of virtual patients. So usually the way I think of them is each of these patients has a unique set of parameters associated with them. So they're all unique or different from each other. And then on these patients, you're then able to test any type of treatment you want and hopefully predict something that's implementable or useful clinically for people who are trying to use this treatment. And so at the backbone of that, or the first thing that we wanted to do then, was calibrate a model. So we wanted to fit the model that I showed you 2 slides ago to data. And so we were very fortunate that we had a good amount of in vivo data. So that was that type of data set where you're growing a tumor in mice. And so these are all not these 9 experiments, tumor volumes in mice grown over time under different permutations of the oncolytic virus. So either VSV, the virus VSV, or the virus VV, and so on. And so we decided to fit this hierarchically, which means that we took subsets of our model and fit those parts first and then built up slowly. So what I mean by that is these 2 experiments here are control experiments, meaning there's no treatment. They're also immune deficient mice, so there's no immune system. So that meant we could ignore all aspects of our model that had virus in it or had immune cells in it, and kind of just delete that in a way and just consider the tumor growth aspects of our model, which I've highlighted in blue. And so that reduced the number of parameters we needed to fit to 3, and we fit those parameters to these data sets. We then consider, oh, there's the image of the flow diagram for what part of the model that spoke to. The next experiment was another control experiment, so no treatment, but in an immune competent mouse, meaning that the mouse had a functioning immune system. So in that case, we were still able to remove all aspects of our model that were about the virus, but had to then consider the immune involvement. So because of that, we fit all the immune related parameters to that data set. And so you can see how we kind of build up slowly all the aspects of our model and fit it to different data sets by using this hierarchical approach. And so after that, we ended up with this fit. I should have mentioned that our fit is in black on each of these plots here, and the data is plotted as a mean and standard error bar. And so from this, we then had a calibrated model that we could create a virtual cohort from. So to create our virtual cohort, we sampled from a normal distribution, which was centered at these parameter values that we'd obtained. And you can see here, this is the distribution of parameter values for our 200 virtual patients. So you can see they all look quite normal. And this is just an example of one of the parameters. So this A1 is the tumor cell cycling rate. So you can see across the population, this was distributed. And so this then means that when you have this kind of distribution of characteristics, you can obtain a distribution of tumor growth, which you can actually see here. This is what our patient cohort's distribution of tumor growth looked like. So some patients had quite slow growing tumors down the bottom here, and some patients had quite rapid growing tumors. And so with this patient cohort, we then wanted to actually investigate those 2 questions. How many injections of the vaccinia or VV virus should we be giving? So we considered that there was a possible number of injections of DP, or we actually thought that DP should just be 7. So a total possible number of VV injections of 7, with our VSV virus given 7 days later. And here what I'm plotting is what the cohort's tumor cell number over time looks like under these 2 different treatments. Either a single dose of VV, so you can see that in blue here, the mean and standard deviation, or 7 injections of VV. So you can see that as a red line for the mean, and standard deviation is in red as well. And so what we noticed for our cohort is that for 1 injection, the variation in the tumor cell number is pretty tight. Well, it's definitely tight in comparison to the 7 injection. What you can see here is that some of the cohort who had the 7 injections actually performed better than when they experienced just 1 injection, which seems to suggest that potentially 7 injections would actually be optimal for some patients. So we decided to investigate that further. And for all of our 200 patients, we optimized their treatment individually, and we determined how many VV injections should they have. So you can see all these green bars here are patients that needed 2 injections. That was their optimal number. So 2 injections of VV was best for them. Whereas all these patients up the top end here, their optimal number of injections was 7. So you can see straight away there's a split in what's optimal for people in our cohort. And we decided to see, is there some characteristic that these patients have that we can identify them by that determines whether they're going to have this response or not? And so overlaid here is the growth of their tumor, or the tumor growth rate. And you can see that patients who needed 2 injections had a much higher growth rate than patients who just needed 7 injections. They have much slower growing tumors. So this suggests that the rate at which a tumor growing would actually determine whether you're going to perform differently under these 2 different treatment options. And so kind of just to summarize that, we did a lot more investigation into this, and we're actually able to determine that the growth rate of the tumor, or the aggressivity of the tumor, will determine what was your optimal treatment, and that patients who had aggressive growth rates actually only needed 1 VV injection, whereas patients with the very slow growing tumors actually needed 7. So this is quite interesting to us because we're able to then see that clinically they're starting to see that in breast cancer with oncolytic viruses, that the aggressiveness of the tumor determines what type of treatment they actually would need and what would be optimal. So this is one example of where we use data. I was going to finish off today by just showing you another example that we've kind of started on, which is actually to do with agent-based models, which is a lot of the talks in this workshop are going to focus on. So Morgan's already kind of introduced what an agent-based model is, but just to give you an overview, there's usually 2 kind of subsets to agent-based models. You either consider them to be lattice-based models, meaning they're kind of fixed to their position in space, or off-lattice models, meaning their position in space can move over time. And here's an example of a tumor growth agent-based model with an oncolytic virus. You can see the infected cells in brown. And this is in PhysiCell, which you're going to learn all about tomorrow, and Paul's going to give a great introduction to. So what I wanted to show you today is where we've been using agent-based models with data. So we've been interested in trying to help optimize what's called a sustained delivery device. So usually a lot of cancer treatments are given intravenously, which means they're injected into the bloodstream. And a lot of the time it's quite difficult for those treatments to actually penetrate the tumor, because you can see tumors have quite poor vascular systems. They're not really good for administering treatment through the veins. So what our collaborators have been investigating is using sustained delivery devices like this one here. And the idea is that you inject the device and it releases the drug slowly over time. So you can see that with the dotted line here. In a way, optimizing the efficacy of the drug, because you keep it localized and you also keep the amount released for longer. And so we have been looking at creating an agent-based model to model that. And we've been using a Voronoi cell-based model. Voronoi cell-based models are just a form of off-lattice agent-based models. And you can see an example of the evolution of our pancreatic tumor model here. So the tumor cells are these orange cells, and the healthy pancreatic tumor, pancreatic tissue is in gray. You can see over time the expansion and the kind of infiltration of the tumor into the surrounding tissue. And so in this off-lattice agent-based model, what we're assuming is that these cells, so if you picked, say, these 2 here, they're actually connected by a spring, just like this one up here. So the way the cells move is that they kind of pull on each other or push each other apart, just like a spring would if you were to take it by 2 ends and push it or pull it apart. And so this is how we kind of formulate movement in the model. And you'll see a simulation of the model in a few slides, and you'll kind of see what I mean by its expansion into the tissue. But the biggest challenge with agent-based models, I feel, is calibrating them to data. And there are a lot of people who are working on different aspects of this, and there are more technical data fitting methods that can be used. So Morgan's going to talk a bit about nonlinear mixed effects modeling later today. Another method that I'm not going to go into, but is useful if you're interested in fitting agent-based models, is approximate Bayesian computation. Also, Bayesian inference can be used. But the one I'm going to mention today is some work I've been doing with Dominique on particle filtering. So the reason agent-based models are difficult sometimes to calibrate is because they're computationally expensive. It's not just like simulating an ODE that usually we can simulate pretty quickly with MATLAB. A lot of the time we create these agent-based models that take time to simulate just one run. So fitting a model isn't necessarily possible with our normal data fitting methods. And so what particle filtering does is it means you can run one single simulation of the model to the data to fit it. And so the idea is that you generate particles. Here you can see in green, I've plotted an example of 50 particles. And what happens is you initialize these particles and their parameter values, and you run them to one data point. And then at that data point, you determine how well did they do? How well did these particles and their parameters do to match the data? And based on how close they were to the data, you then weight them. Given their weighting, you then resample the particles. And so your likelihood of choosing a particle that did well is very high. And then you also perturb slightly their parameter values. So you kind of add a little bit of noise to those parameter values they originally had, and then you continue to the next data point. And you keep doing this as your data evolves. And the idea is that you end up with a calibrated distribution or a set of parameter values for your agent-based model. So you can see here, these kind of is what our results came out like. So I should have mentioned that the gray is our data. So it's, again, in vivo mice tumor volume over time. And the green lines are the Voronoi cell-based model, or the agent-based model you saw on the previous slide. And so this method was actually really useful to us because our model was computationally expensive to simulate. So it allowed us to run one simulation and calibrate a set of parameters. And then from that, I've been working with a student, Isabel here at QUT, who's been helping me now to actually investigate. Can we help understand whether these sustained delivery devices can be optimized? And so the first thing we've been doing is a multi-parameter sensitivity analysis to determine what's sensitive in this agent-based model. We were able to determine, maybe not surprisingly, but the age at which a cell differentiates is a very sensitive part to determining a tumor volume. And then we've also been looking at what happens when you inject this drug, the anti-cancer drug, into different locations. Do we see that different locations are optimal over others? And our preliminary results at the moment are currently that we're seeing that 4 injections spread throughout the tumor is optimal over just a single intratumoral injection. You can see that here by looking at, over time, the number of tumor cells in the model. So we're currently working on the sustained delivery device and looking at what the optimal delivery from these devices should be. And we should hopefully have some very interesting results to come on that. So I just wanted to mainly keep that part in there because I thought it was quite interesting. Talking about data fitting with agent-based models is a big challenge. And hopefully we'll see a lot more talks this week on agent-based models, and we'll get to discuss different data fitting methods or different data calibration methods. So that is everything for me. Of course, I just want to thank all the collaborators. A lot of people work on these projects I'm part of. I don't just get to do it all myself. And also thank all of you for attending today. And I'm really looking forward to the rest of today. I think we've got a great lineup of speakers to come. And yeah.
Thanks, Adrianne. So it's great to see where the sustained delivery device has gone. I haven't heard about that in a while, so it's fantastic work. There are a number of questions. I think some you and I can handle via chat, but I'll just ask a few. This is from Miranda Lynch. For the virtual cohort modeling, can you comment on the role of chosen sample sizes for the cohort? How do you determine how many samples to simulate? How many data are needed to support the hierarchical model parameter estimation?
So I think this is a really good question. I actually gave a similar talk about a month ago, and someone asked me pretty much the same question. And I've thought of it in the sense that we currently show the result for 200, and I've simulated 400 patients, and there was no real difference. So I figured that 200 was a good number. But there is actually a way of determining what the kind of lowest best number is for your virtual cohort. And someone actually told me about it when I gave this talk a while ago. I can't remember offhand, but there is a way of inferring from the variation in your model that comes out from your virtual cohort, roughly how many patients you're going to want to simulate to make sure you're getting a robust or reliable prediction. So for us, we kind of felt like 200 hit a good mark. We felt like our results were robust because as we increased the number of virtual patients, it didn't really seem to impact our results. But there is a way to actually do this and to infer from variation what it should be. And I'm going to start to look into it now with this person who asked this question in my last talk. I'm just looking at the second part of your question. How do you determine how many samples to simulate and how many data are needed to support the hierarchical? Yeah, so I think how much data is needed to support hierarchical model parameter estimation is a really good question. I think that what I showed today was using a least squares nonlinear fit, which is that very simple fitting method I showed at the beginning. However, we can do much better than that. we don't necessarily gain any more information by doing a Bayesian inference approach, but by doing that, we can double check that the parameters that were obtained are identifiable and actually reliable estimates. For us, when we fit the data, we fit a max of 3 parameters to each of those data sets. And because the data sets each contained, I think, about 12 data points, and each of those average 12 data points contained 4 mice or more, we felt that there was sufficient data in that case to fit the parameters. But I would be hesitant to fit any more than that to those data sets there. So that would kind of be my answer, which is not a rigorous answer, but
I think it's always somewhat heuristic, right? Like it depends on the quality of your data, it depends on what aspects of your model you're fitting and how those parameters together. So, okay, let's maybe pass the next question, and then we'll take a break, and you and I can handle some of the questions in the chat. This is a question from Melissa Stadt. I hope I said your last name correctly. In the virtual populations, are there ways to consider differences between male females? For example, if there are any sex differences in the specific cancer? I think that's a great question.
I think that is a fantastic question. So we've been throwing around this idea for some time now about trying to extrapolate a virtual population that's based on gender or kind of demographic characteristics that can help inform like how treatment might perform differently. I think it all comes down to what data you have available. So absolutely, you could definitely consider how this treatment might act on a female cohort versus a male if you had reliable data sets that could actually inform the characteristics for your virtual population. So we haven't done that. The example I gave today was for triple negative breast cancer, which predominantly occurs in women. So I guess our cohort, if we tried to kind of think about what the identity of the patients were, it would be predominantly females, but we didn't do any kind of gender identification. There is a way to create virtual cohorts by sampling from body weight and looking at how drug distributes through the body based on body weight, and then trying to figure out how to optimize treatment a bit more individual, like a bit better for people individually that way. But we haven't done it, but I think it's a really great question, and that someone should definitely do it.
And Kristen Swanson at the Mayo Clinic looks a lot at those kinds of models. So if you're interested in that, I think at the Mayo Clinic. So I think she would be a wonderful resource. And yeah. All right. Well, it is 10:01. So let's try and stick to the schedule as much as we're going to be able to. And there are questions in the chat. I'm sending out an answer to Thomas now, and then maybe you could handle some of the rest of the ones for your talk. Though I do see one question, can delayed differential equations be used? Yes, that's actually what we use. So that's a great question. The next speaker at 10:30 a.m. Eastern time is Lizette DePillis, and again, terrible name pronunciation. So she'll be joining us at 10:30. So we'll have a half an hour break, and we'll meet back for the afternoon and the keynote sessions. So thanks very much, everyone.